Hello, welcome to Biology Descent Study Club. Plant development, cell signaling, and transcriptional regulation. The genetic analysis of a plant development using model organisms such as Arabidopsis has lagged behind that of animal models simply because there are fewer researchers working on plants. For example, in 21, when the Arabidopsis DNA sequence was completed, fewer than 5% of its gene had been defined by mutational analysis, whereas over 25% of the genes in both Drosophila and C. elegans had been identified in that way. We are just beginning to understand the molecular basis of plant development in detail. Thanks to DNA technology and clues from animal research, plant research is now progressing rapidly. Mechanisms of plant development. In general, cell lineage is much less important for pattern formation in plants than in animals. As mentioned previously, many plant cells are totipotent, and their fates depend more on positional formation than on cell lineage. Therefore, the major mechanisms regulating development are cell signaling, induction, and transcriptional regulation. The embryonic development of master plants occur inside the seed, and thus is relatively inaccessible to study. A mature seed already contains a fully formed embryo. However, other important aspects of plant development are observable throughout a plant's life and its meristems, particularly the apical meristems at the tips of shoots. It is there that cell division, morphogenesis, and differentiation give rise to new organs such as leaves or the petals of flowers. We will discuss two aspects of pattern formation in floral meristems: the apical meristems that produce flowers, pattern formation in flowers. Environmental signals such as day length and temperature trigger signal transduction pathways that convert ordinary shoot meristems to floral meristems, causing a plant to flower. Researchers have combined a genetic approach with tissue transplantation to study induction in the development of tomato flowers. Notice this figure. A floral meristems in a bomb consisting of 3 layers of cells. Notice that L1, L3. Notice that cell layers. All 3 layers participate in the formation of a flower. Reproductive. Notice that. Structure with 4 types of organs: carpels containing egg cells, stamens containing sperm-bearing pollen, petals and spillas leaf-like structures outside the petals. In a mature plant, the 4 types of organs are arranged radially rather than linearly like the body structures in Drosophila. Tomato plants homozygous for a mutant allele called fascinated F produce flowers with an abnormal large number of organs. To study what controls the number of organs, researchers performed the grafting experiment outlined in this figure. Notice that F. The grafted stems from fascinated plants into those of wild type plants, FF homozygous for normal allele. Notice that. And then grow new plants from the shoots that supported near the graft sites. Many of the new plants were chimeras or organism with a mixture of genetically different cells. Some chimeras produced floral meristers in which the three cell layers did not all come from the same parent. The researchers identified the parental sources of the meristem layers by monitoring other genetic markers such as an unrelated mutation causing yellow leaves, or research notice that or notice also the results. The results showed that whether the number of floral organs was normal or abnormal high depended on whether the L3 layer arose from wild type or mutant cells. Thus, the L3 cell layer induced the overlying L2 and L1 layers to form a particular number of organs. The mechanism of cell-cell signaling leading to this induction is not yet known, but is currently under study. In contrast to genes controlling organ number in flowers, are genes controlling organ identity. An organ identity gene determines the type of structure that will grow from the meristems. For instance, whether a particular outgrowth from a floral meristem becomes a petal or a stamen. Most of what we know about such organ identity genes comes from research or on flower development in Arabidopsis. Organ identity genes are analogous in homeotic genes in animals and are often referred to as plant homeotic genes. Just as mutation in a fruit fly homeotic gene can cause legs to grow in a blast of antenna, a mutation in an organ identity gene can cause crabbles to grow in a blast of spells. By collecting and studying mutants with abnormal flowers, researchers have been able to identify and clone a number of floral notice also floral organ identity genes in plants with a homeotic mutation. Specific organs are missing or repeated. Some of these mutant phenotypes are reminiscent of those caused by bicoid or other pattern formation mutations in Drosophila, like the homeotic genes of animals. The organ identity genes of plants encode transcription factors that regulate specific target genes by binding to their enhancers in the DNA. Notice also back to inquiry which cell layers in the floral meristem determine the number of floral organs. Experiment tomato plants with the fascinating FF mutation develop extra floral organs. Notice that. Notice wild type, normal, and fascinated F extra organs. Notice the results. The flowers of the cameric plants had a fascinating phenotype only when 3 layers came from the fascinated parent. Notice that. Notice also the wild type parent, and notice camera and camera 1, 2, 3. Notice the differences here. Back to mutations in floral organ identity genes. Notice the differences here. Wild type, this is represent wild type Arabidopsis has 4 sepals and 4 petals, 6 stamens, and 2 carpels. If an organ identity gene called abetala2 is mutated, the identities of the organs of the 4 walls are carpel, stem,